Welcome to Last Minute Lecture. This free chapter overview is designed to help students review and understand key concepts. These summaries supplement, not replace, the original textbook and may not be redistributed or resold. For complete coverage, always consult the official text. Usually when we talk about the global energy grid, the conversation immediately goes to massive infrastructure. We picture solar farms stretching for miles or deep offshore oil rigs. We tend to think of energy as a strictly human-engineered domain.
Right. Well, it's natural to assume that. We see the big machines, so we assume energy is a purely macro-level operation.
But then you dive into the microscopic world specifically, the domain of life known as Archaea, and suddenly that whole picture just shifts completely. Because long before politicians were arguing about energy security, these invisible organisms were, busy generating methane, which is exactly what we harvest as natural gas. So for you listening right now, you are about to embark on a deep dive into the complex, frankly alien biology of Archaea. Our mission today is to take Chapter 19 of Prescott's Microbiology and translate these dense microbial mechanisms into clear, memorable concepts so you can master this material for your microbiology studies.
Yeah, and setting the stage with that natural gas connection, it really grounds these microscopic organisms in our everyday reality. The United States alone has roughly 2,300 trillion cubic feet of recoverable natural gas, and much of it is locked deep underground in shale rock formations. All of that is essentially ancient biologically produced methane.
That is a massive catch, though. Extracting that gas requires horizontal drilling and hydraulic fracturing, what we all know as fracking. You're pumping millions of liters of water, sand, and chemicals under extreme pressure to crack open the shale.
Which, of course, introduces significant environmental considerations. The process is associated with potential groundwater contamination, fracking-induced earthquakes, and uncontrolled methane leaks from the well sites. To be clear, our goal here isn't to weigh in on the politics or ethics of energy policy. We're simply impartial observers of the science here.
Exactly. We're just looking at the facts.
Right. But understanding that context frames Archaea perfectly. They aren't just strange oddities living in the dirt. They are organisms directly powering and, complicating our modern world.
If their impact on our world is that massive, we really need to understand what an archaeon actually is from the ground up. Because the deeper you look into their biology, they come across as this bizarre hybrid. They almost seem cobbled together from completely different branches of the tree of life.
That hybrid nature is what makes them so unique. If you look at their informational processing, meaning how they replicate their DNA or the machinery they use to transcribe and translate that DNA into proteins, they operate incredibly similarly to eukaryotes. Eukaryotes being, the complex cells that make up plants, animals, fungi, and humans.
Yet when you look at their metabolism, how they actually scrounge for food and generate energy, they behave far more like standard bacteria.
Yeah.
But we run into a major roadblock when we try to study that metabolism.
Yeah.
There is this looming issue of microbial dark matter. Most Archaea have never been successfully grown in a laboratory petri dish.
Right. We simply don't know how to replicate the extreme or highly specific conditions they need to survive. So instead of growing them, scientists rely on culture-free methods like metagenomics and single-cell genomic sequencing. We are essentially scooping up soil or deep-sea water, extracting whatever DNA is in there, and inferring how these organisms live entirely by reading their genetic blueprints.
We're reading the architectural plans without ever seeing the physical building.
Exactly.
Let's look at those blueprints, starting with how they build themselves from scratch through carbon fixation, or autotrophy. They use carbon dioxide to build complex organic molecules, and there are three major pathways to do this. I want to start with the Wood-Ljungdahl pathway because the mechanics of it are fascinating, especially when you look at figure 19.1 in the text.
It is an incredibly elegant system, mostly because it is the most energetically favorable route available. Picture a microscopic branched assembly line. On one branch, a carbon dioxide molecule is captured and reduced down to carbon monoxide.
Okay, simple enough.
Yeah, and on a completely separate branch, a second Ca2 molecule is captured, reduced, and chemically bound to a specialized carrier molecule called tetrahydromethanopterin.
So you have these two separate conveyor belts running simultaneously.
And then they merge. An enzyme complex grabs the carbon monoxide from the first belt and the single carbon from the second belt and fuses them together. The result is a crucial two-carbon molecule called acetyl-CoA, which the cell then uses as the fundamental building block for everything else it needs.
And the best part for the Archaean, running this entire dual conveyor belt only costs one single molecule of ATP.
Right, it's just one ATP.
That is an unbelievable level of efficiency. But that's not the only way they fix carbon. There are two other major cycles to compare, the HPHP cycle and the DCHP cycle. When you put them side by side, like in figure 19.2, the energy costs are wildly different.
Oh, the contrast is stark. The HPHP cycle is an absolute energy hog. It costs the cell 9 ATP for every molecule of pyruvate it manages to synthesize. The DCHP cycle, on the other hand, is much cheaper, costing only 5 ATP.
Why would any organism willingly use a pathway that costs almost double the energy? That feels like an evolutionary disadvantage.
Well, it all comes down to oxygen. The enzymes running the cheaper 5 ATP DCHB cycle are highly sensitive to oxygen. They will literally fall apart if exposed to it.
Oh, wow. So they can't be around air at all.
Exactly.
Yeah.
The expensive HPHB cycle, however, is completely oxygen tolerant. So if an archaeon happens to live in an aerobic, oxygen-rich environment, it has no choice. It is forced to pay that heavy 9 ATP price tag just to survive the surrounding chemistry.
It's essentially a massive tax on breathing oxygen. Speaking of metabolic taxes, there's another pathway that caught my attention, figure 19.3. Certain salt-loving Archaea, the Heterotrophic, use something called the methyl aspartate cycle to process carbon.
Right.
When you map this cycle out, it involves significantly more chemical steps than the standard glyoxylate cycle used by other microbes. More steps usually means more enzymes to build and more energy wasted. Why evolve such a complicated detour?
That detour is actually a highly specialized survival mechanism. Archaea live in environments where the food supply is incredibly unpredictable. When nutrients are abundant, they gorge themselves, storing excess carbon inside their cells as massive, dense polymers called PHA, essentially a biological plastic.
Like a microscopic pantry for a rainy day.
Exactly. But when starvation hits and they need to break down that stored PHA plastic, using a standard, simple metabolic cycle would cause a massive chemical traffic jam. The intermediate byproducts would build up faster than the cell could process them, becoming toxic. Yeah, so the longer, more complex methyl aspartate cycle bypasses that traffic jam entirely. It's a specialized chemical detour designed specifically to unpack those dense PHA stores safely when the environment turns hostile.
So the complexity is the point. It's a pressure release valve. Now, taking this idea of extreme survival to its absolute limit, we have to talk about how some of these organisms break down sugars like glucose. They use a modified Embden-Meyerhof pathway, shown in figure 19.4, that yields exactly zero net ATP.
Zero net ATP.
Yeah. Think about running a business where every single transaction results in zero profit. How do you stay alive when your energy yield is zero?
It totally defies our standard understanding of biology. But they pull it off by substituting standard biological molecules for high-octane alternatives. For instance, instead of using the common electron carrier NAD plus A, they use ferredoxin, which holds significantly more reductive power.
Why do they have to do that?
Because many of these archaea live so deep in the earth or in such extreme conditions that they operate right at the absolute thermodynamic limit of life.
Meaning there is barely enough ambient energy for the laws of physics to allow life to exist at all.
Right. They have absolutely no ATP to spare for inefficiencies. Their entire internal chemistry has been stripped down and re-engineered for break-even, ultra-efficient survival rather than bulk energy production.
Surviving on a razor's edge. Now, if their internal engines are so alien, where do they actually fit on the family tree of life? Do they even belong? This brings us to a mind-bending group called the Asgardarchaeota.
Yes, the Asgardarchaeota.
They are named after the realm of the Norse gods because they were discovered hidden away in deep, remote marine sediments. What researchers found in their DNA fundamentally shook biology.
It really did. Their genomes encode what we call eukaryotic signature proteins. For decades, scientists believed these specific proteins were exclusive to complex cells, plants, animals, us. We're talking about actin and tubulin, which form the dynamic internal skeleton of a cell, and ESCRT proteins, which are used to bend and remodel complex cellular membranes.
Let me push back on that, though. When researchers analyzed one specific cultured species from this group, Candidatus prometheor camus and trophicum, they found it possessed the genetic codes for intracellular trafficking the proteins that shuttle materials around inside a cell.
Yeah, that's right.
But the organism itself has no internal organelles. It has no internal compartments. Isn't that like building an intricate multi-lane highway system and hiring a bunch of traffic cops in an empty field with absolutely no buildings? What on earth are they trafficking?
That empty field analogy is spot on, and it points to a profound evolutionary mystery. The prevailing theory is that these organisms represent a living snapshot of the deep past. They are currently considered the closest living relatives to the ancestral host cell that, billions of years ago, swallowed a free-living bacterium, an event that created the very first eukaryotic cell.
Oh, wow.
Yeah, they possess the complex genetic toolkit for cellular highways, even if they aren't fully utilizing them to shuttle materials between organelles yet.
So they are carrying the ancient blueprints for our own cellular biology. Alongside these evolutionary ghosts, we find another fascinating group, the nanoarchaeota. And as the prefix nano suggests, they are unbelievably small.
Barely 0.2 micrometers in diameter. Their genomes are heavily reduced, meaning they have completely lost the genetic instructions for fundamental survival mechanisms. For example, they completely lack an electron transport chain. They physically cannot generate their own energy.
So they are obligate symbionts. They are forced to latch onto a host just to survive.
And we can see exactly how this plays out in some of the most hostile environments on Earth. Consider the thermoproteota, a group that thrives in sulfataras.
For you listening, a sulfatara is a geothermally heated pool, like a volcanic hot spring. But they are milky white, and that milkiness is actually a massive concentration of elemental sulfur.
In those boiling sulfurous cauldrons, we find organisms like Pyrolobus fumaria. Its optimum temperature for simply going about its day is 105 degrees Celsius. Researchers literally put this organism inside a laboratory autoclave, the high-pressure steam machines we use to sterilize medical equipment, and it easily survived an hour at 121 degrees Celsius.
It treats our ultimate sterilization tool like a warm bath.
Exactly. And within these hydrothermal environments, we find a perfect real-world example of this tiny nanoarchaeota we mentioned earlier. If you look at figure 19.6 in the book, which shows a confocal microscopy image, you'd see tiny red parasitic cells of Nanoarchaeum equitans physically clinging to the outside of much larger green host cells, a species called Ignococcus hospitalis.
How does that microscopic heist actually work? How does the tiny parasite steal the energy?
Well, it's all about how the host, Ignococcus hospitalis, is built. Unlike most Archaea, it lacks a rigid outer protein shell. Instead, it has two completely separate membranes. Remember that oxygen-sensitive DCHB carbon fixation cycle we discussed?
Yeah, the one that costs 5 ATP.
Right. The host runs that cycle, but it produces highly toxic aldehydes as a byproduct. So the host hides the enzymes for this cycle in the space between its two membranes.
It builds a biological containment room to protect its own DNA from its toxic factory.
Precisely. But to power that factory, the host's outermost membrane is packed with ATP synthase, the cellular engine that generates energy. The tiny parasite, N. equitans, takes advantage of this architecture. It physically plugs itself directly into that energized outer membrane and siphons off the ATP before the host can even use it. It's capping directly into the power line. If we're talking about extreme architectural adaptations, we have to look at figure 19.8 and discuss Saccharolobus sulfataricus. It's a thermoacidophile. It grows happily at a pH between 2 and 4. Now, pH 2 is essentially the acidity of battery acid.
It is incredibly acidic.
Yeah, but internally, its cytoplasm is maintained near a neutral pH of 6.5. If the outside is battery acid and the inside is neutral water, the cell is literally functioning as a biological battery, isn't it?
It absolutely is. The laws of chemistry dictate that the massive concentration of protons, the acid on the outside, desperately wants to rush into the neutral interior to balance things out.
Right, diffusion.
Yeah, and the Archaeon uses that intense rushing river of protons to spin its ATP syntheses, generating power. And it goes further than that. It leverages that inward acidic current to run at least 15 different secondary transport systems. It uses the physical force of the inward flow to drag essential nutrients, like sugars and amino acids, into the cell against their natural concentration gradients. It turns a hostile acidic environment into an unlimited power grid. Now, after surviving boiling acid and hydrothermal vents, you'd be forgiven for thinking every Archaeon is an extremophile. But they aren't. We find them in everyday environments, too. Let's look at the nitrospheria, which populate our standard soils and oceans.
These are mesophiles, organisms that thrive at moderate everyday temperatures. They play a massive role in global nutrient cycling as mixotrophs. They pull energy by oxidizing ammonia into nitrite using an enzyme called AM, while simultaneously absorbing organic carbon from the surrounding environment to physically build their cellular structures.
And to survive in cooler environments, they have a very specific architectural adaptation in their cell membranes, a lipid molecule called Archaeal. What's critical here, as shown in figure 19.9, is that Archaeal contains a unique cyclohexane ring, a structure with 6 carbon atoms. Now, the hyperthermophiles we talked about, the ones in boiling water, use cyclopentane rings, which only have 5 carbons.
Right.
Why does a single carbon atom make such a difference?
It dictates the physical fluidity of the organism's outer boundary. Hyperthermophiles live in boiling water. If their membranes aren't incredibly rigid, they will literally melt and dissolve. The 5-carbon cyclopentane ring provides that extreme rigidity.
Okay, makes sense for boiling water.
But mesophiles live in cool oceans. If they use that same rigid armor in cold water, their membranes would freeze solid and shatter. Evolving that 6-carbon cyclohexene ring prevents the lipid molecules from packing too tightly together. It guarantees the membrane stays fluid and flexible at lower temperatures.
So their surrounding temperature dictates their molecular shape down to a single carbon atom. All right, let's bring this journey full circle. We started this deep dive talking about the natural gas that powers our cities. Let's finally meet the architects of that gas, the methanogens.
Methanogens are strict anaerobes. The slightest whiff of oxygen is completely toxic to them. To generate methane, they utilize a highly complex biochemical pathway known as the Wolff cycle, which is detailed in figures 19.10 and 19.11.
The Wolff cycle looks like an absolute nightmare of chemistry when you see it mapped out. But if we translate it, it's basically a microscopic relay race, where a single molecule of carbon dioxide is passed like a baton from runner to runner.
And each runner is a highly specialized coenzyme. The CO2 enters the cycle and is handed to the first coenzyme, methanofuran. Then it gets handed off to tetrahydromethanopterin, then down to coenzyme M, and finally to coenzyme B.
So it's just jumping down the line.
Exactly. At each stage of this relay, helper molecules like ferredoxin and a unique coenzyme called F420 strip oxygen atoms away from the carbon and hammer hydrogen atoms onto it. Step by step, they chemically force that CO2 to become methane, CH4.
But there's the trap at the end of the race. The final chemical reaction releases the methane gas, but it leaves the last 2 runners, coenzyme M and coenzyme B, physically fused together in a rigid disulfide bond.
The cell has to pry them apart to run the cycle again, which requires a lot of energy. And to crack that bond, they use an astonishing mechanism called flavin-based electron bifurcation. It works almost like a biological seesaw. The cell takes a pair of incoming electrons and splits their paths. It drops 1 electron down an easy energy-releasing pathway.
Okay.
It then uses the mechanical momentum from that drop to launch the second electron up a highly difficult, energy-requiring pathway. This forceful split shatters the disulfide bond, recycles the coenzymes, and resets the chemical trap for the next CO2 molecule.
And the sheer scale of this microscopic relay race is hard to fathom. Methanogens produce roughly 1 billion tons of methane globally every single year. Just the methanogens living inside the rumen of a single dairy cow release over 200 liters of methane a day.
Which is deeply concerning for global warming, given how potent methane is as a greenhouse gas. However, Archaea also provide the counterbalance. There is a group of methane-oxidizing Archaea, specifically a lineage called ANME, that actually saves us from a methane-choked atmosphere.
By doing the exact opposite.
Right. They run the entire Wolff cycle in reverse. Instead of producing methane, they consume it from the environment. But to oxidize that methane, they have a massive excess of electrons they need to dump. They solve this by forming a tight physical partnership with sulfate-reducing bacteria. If you viewed them under a microscope, like in figure 19.12, you would see a core of these Archaea physically wrapped in a thick shell of bacteria.
And they share resources.
They share electricity. The Archaea physically pass their excess electrons directly into the surrounding bacteria using conductive pili, which function exactly like biological microscopic nanowires.
That is science fiction happening in the mud. Let's look at one final extreme group before we wrap up the Archaea. These thrive in hypersaline environments, like the Dead Sea, or those coastal salterns that turn brilliant, opaque red from the microbes' pigments, as seen in figure 19.32. Basic biological physics dictates that if you place a normal cell into water that is 36% salt, osmosis takes over. The water violently rushes out of the cell to dilute the surrounding salt, and the cell instantly shrivels up and dies. How do Archaea beat osmosis?
They employ a counterintuitive salt-in strategy. Rather than fighting to keep the salt out, they embrace it. They use molecular pumps to aggressively pull massive amounts of potassium and sodium ions from the environment directly into their own cytoplasm. They pump salt inward until their internal concentration perfectly matches the extreme external environment.
But wait, if a cell is completely flooded with salt, standard proteins will denature and map. They literally unravel and stop working entirely.
True, which is why Archaea fundamentally re-engineered their entire protein structure. The outer surfaces of their proteins are heavily coated in acidic amino acids. These highly acidic shells act like molecular magnets for water. They tightly grip onto a microscopic layer of water molecules, pulling a protective hydration shell around the protein.
Oh, that's clever.
Yeah, this shell repels the surrounding salt and allows the protein to fold and function perfectly.
And on top of that, they synthesize a unique protein called archaeodopsin. This protein uses a molecule called retinol, which is the exact same light-sensitive pigment we use in the retinas of human eyes to catch sunlight.
The mechanism is beautifully simple. When a photon of sunlight strikes the retinol, the molecule physically bends and changes shape. That mechanical movement pushes a single proton across the cell membrane. By doing this thousands of times, they create a proton gradient to spin their ATP synthesis. They generate power directly from the sun, entirely without using the complex chlorophyll machinery that plants use.
Life simply refuses to be boxed in. And just to drive that point home, our source material briefly notes organisms like thermoplasma, which thrives in the hot, toxic refuse piles of coal mines at a pH of 1 to 2 and manages to survive without even having a cell wall, and Picrophilus, an organism that can actively grow at a pH of exactly 0.
They redefine the absolute limits of biochemistry. Archaea show us that wherever there is even a fraction of usable thermodynamic energy, biology will find a way to harness it.
So take a breath and think about the journey we just took. We started with organisms running 0 ATP engines just to scrape by in the dark. We met the Asgardarchaeota, harboring the genetic blueprints of our own complex cells. We witnessed microscopic heists and single cells functioning as biological batteries in boiling acid. We learned how a single cyclohexane ring keeps mesophiles from freezing. And we explored the methane makers and the methane eaters connected by nanowires that physically shape the climate of our planet.
It is an overwhelming amount of biochemical ingenuity packed into a single domain of life. And looking at all this leaves us with a fascinating, somewhat philosophical question to ponder.
Lay it on me.
Think back to the Asgardarchaeota and their eukaryotic signature proteins. If a member of this group is successfully cultured, and researchers crack it open and confirm it contains functioning internal membranes, and if the genetic evidence holds that they are the direct ancestors of the original eukaryotic host cell.
Well, biologically speaking, should eukaryotes just be reclassified as a highly derived, specialized branch of Archaea? Meaning, are you, me, and everything we see around us technically just highly evolved Archaea?
It completely blurs the tree of life. It reminds us that the rigid boundaries we draw in biology are often just temporary human constructs, waiting to be dismantled by the next big discovery hidden in the microbial dark matter.
That is a profound thought to let linger. Something to chew on the next time you look in the mirror or the next time you ignite the burner on your gas stove. Thank you so much for joining us on this deep dive into Chapter 19 of Trescott's Microbiology. This deep dive was brought to you by the Last Minute Lecture Team. We wish you the absolute best of luck mastering this material for your microbiology studies. Keep questioning the invisible world around you. Keep learning, and we'll catch you next time.